There are few aromas in this world that give me as much pleasure as the smell of freshly ground coffee beans. Though that hasn't always been the case. You could say it's an acquired smell, but it's not just the olfactory cues that my mind is reacting to. It's also the anticipation of a reward in the form of a boost in focus and arousal. I fully admit that I have a pretty strong addiction to the most popular psychoactive drug in the world, caffeine. Caffeine has a number of effects on the body, but perhaps the one that keeps us coming back is its effect on a type of receptor in our brain that is triggered by a molecule called adenosine. When adenosine binds to this receptor, like a key inserting into a lock, it sets off a series of reactions whose ultimate effect is largely to inhibit or turn down the activity in the central nervous system. Adenosine is made when our cells are active. You might already be familiar with ATP, adenosine triphosphate, which is the fuel that our cells run on. When they need energy, they break down ATP, like combusting gas, and what's left is a series of byproducts of metabolism, including adenosine. So as your cells use fuel, more and more adenosine builds up in the nervous system. When it binds to adenosine receptors, this signals the cells to slow down their energy use, encouraging them to take a rest. When you've got lots of adenosine buildup, you start to feel sleepy. Time for a nap or a coffee break. Why coffee? Because caffeine blocks adenosine receptors. It prevents them from accepting adenosine, but doesn't activate the inhibitory effect. This process also leads to more of the neurotransmitters dopamine and glutamate exerting their influence on the brain. You not only feel less sleepy, but you also get a burst of energy and focus. And I'll admit that these lectures were written when I was under the influence of some pretty great coffee. It's pretty amazing to think that you can go from one state of mind in which your body is pushing you towards rest to its complete opposite, jittery, focused, fully awake, just by drinking a shot or 2 or 6 of espresso. So does that mean that our brain function, our state of mind, and everything else that controls our behavior depends upon the relative concentrations of chemicals floating around in our cerebrospinal fluid? Is it accurate to say that a person who suffers from a mental disorder simply has a chemical imbalance in their brain? Given the powerful role that neurotransmitters play in our minds, it's easy to see how this explanation has become so compelling. But it's a myth that the major psychiatric illnesses stem from too much or too little of one neurotransmitter. I wish it were as simple as that. Where did this notion come from? Well, we can trace its roots to some pretty remarkable findings, often serendipitous ones, when doctors observe profound effects on a person's psychiatric well-being with the administration of a single drug. Before the discovery of antipsychotic drugs, for example, doctors had virtually no effective treatment options. But with a few pills or tinctures, a patient with a tenuous hold on reality could suddenly seem calm and rational. These dramatic effects suggested that there was a fairly straightforward relationship between mental illness and neurochemistry. As a case in point, John Cade was an Australian psychiatrist who thought that bipolar disorder, in which patients alternate between states of mania and depression, resulted from too much of one substance during the manic episodes and too little of the same thing during depressive episodes. Believing that the substance in question could be found in urine, he injected guinea pigs with urine from patients in the manic state and urine from healthy people. And he found that the urine from manic patients was more likely to kill the guinea pigs. So he wanted to figure out what was toxic about manic patient urine, and to do so, he injected the pigs with lithium urate, thinking that was the toxic compound. But instead of getting sicker, the pigs seemed to chill out. They became calm and lethargic. And so he wondered whether lithium might also calm down his patients who suffered from mania, schizophrenia, and depression. Sure enough, the substance seemed to abate psychotic excitement, particularly in patients who were in the throes of manic episodes. Lithium is still widely prescribed for the treatment of bipolar disorder, largely because it seems to reduce the manic state. It also might help prevent suicide. But while we know it interacts with other neurotransmitters implicated in mood disorders, we still don't really know how or why it works. A similar situation led to the discovery of other antipsychotic medications, the first being chlorpromazine. Initially, a precursor of the compound was tested for treatment of infection in patients who were awaiting surgery. But it wasn't effective. It did seem to calm them down, though. So a surgeon in the French Navy asked for some samples of chlorpromazine to reduce anxiety that can sometimes be provoked by anesthesia. It proved to be very effective, and the surgeon then wondered whether it might calm patients with mental illness as well. Sure enough, within 5 years, the drug was found to be helpful in patients with schizophrenia and other causes of psychosis. This discovery led to a shift in the treatment of psychiatric disorders, away from the hospitals and into the psychiatrist's office. It was so successful that pharmaceutical companies began searching for similar compounds that they could then bring to market. One result of this search was the discovery of a class of antidepressants still widely used today. While searching for a chlorpromazine-like compound, a Swiss psychiatrist tested the first tricyclic antidepressant on his patients with schizophrenia. It didn't help them. But before tossing it out, he tried it on a patient with severe depression. And a whole new class of drugs used to treat depression was born. In this case, the first being imipramine with the brand name Tofranil. With so many, if not all of these drugs, the mechanisms by which they work either remain somewhat mysterious still, or were only discovered after they were shown to work. This process is a little bit like reverse engineering. Imagine that someone handed you a smartphone and asked you to figure out how it works. What would you do? You might eventually figure it out, but you'd likely also take a lot of wrong turns along the way. And the manual you would write to explain its workings would look very different from the one written by the engineers who built and designed it in the first place. This is what is happening right now in neuroscience. We're trying to reverse engineer the brain with a limited set of tools to query how it works. And we're likely missing some pretty important aspects. Mental illness is complicated, devastating, and stigmatized. So it's no wonder that we want to simplify it as much as possible. I wish depression were as simple as an imbalance of serotonin, or schizophrenia was just about too much dopamine. Because then we could fix the problem much more easily. But it's just not that simple. So let's get into the details that we do know. First of all, there is one major difference between the way that chemicals enter the brain and how they roam around the rest of the body. And that's because what's good for the body isn't always good for the brain. Your body has a built-in protection system. When foreigners invade, it unleashes an immune system response to fight them off. The immune system works in a number of ways, but one crude defense mechanism is to kill infected cells. But if a bacterial colony infected your brain cells, and the immune system's response was to kill them off, you'd have a big problem. Instead, we've evolved what's known as the blood-brain barrier, a physical barrier between the blood circulating in the rest of our bodies and our brain. Endothelial cells line the capillaries in the brain, preventing most compounds from moving between the blood and the cells of the brain. Molecules like oxygen and hormones are small enough to pass through, but most bacteria, viruses, and toxins are kept out. This barrier ensures that bacterial infections of the brain are very rare, though when they do happen, they can be very serious. But it also poses a problem for drug companies that want to alter the balance of chemicals in your brain. You can't just ingest a pill and expect that compound to make its way into the brain. Getting past the blood-brain barrier is a major challenge for pharmaceutical companies. Caffeine gets through because it's so similar to adenosine, which, as we learned about earlier, is found throughout the nervous system. Alcohol passes through too, because of its electrochemical properties. And luckily, many antidepressants and other psychiatric medications contain compounds that are small enough to get through, but not all of them. Along the blood-brain barrier, there are specialized gates that let in specific larger compounds that are useful to the brain. But still, most pharmaceutical compounds out there simply get left out. Once a compound does make it through, it then needs to actually affect the functioning of the brain. In our quest to reverse engineer the brain, neuroscientists have focused on how neurotransmitters, or the chemicals that are involved in signaling between neurons, work. Remember that dendrites are the parts of neurons that receive information, while axons are the projections that pass on the signal. Both axons and dendrites contain gates along their membranes that selectively allow certain compounds to pass through. Along the dendrites, these gates are called receptors because they're receptive to specific neurotransmitters. These neurotransmitters generally come from the ends of axons from the upstream neurons and are released when an action potential, a little electric wave, makes it to the axon's terminus. The neurotransmitters are then released into the synapse, the space between the upstream axon and the downstream dendrites. And at that point, simple diffusion disperses the neurotransmitters, and some proportion of them bump into receptors. Now, neurotransmitters can affect receptors in different ways, but one common effect is to cause the receptor to open a gate that changes the proportion of negative and positive ions inside the neuron. Remember that the inside of the cell is slightly more negative than the outside. So positive ions outside the cell are hankering to come into the cell. When the gates open, they can do so. This influx causes the inside of the cell to become less negative, or slightly more positive. And if enough gates are opened, the inside of the cell becomes positive enough to pass a threshold and set off an action potential down its own axon, which causes it to release neurotransmitters at the terminus and so forth, all along the complex circuitry of the brain. Now, some neurotransmitters actually block gates, making it less likely that the neuron will send an action potential. And neurotransmitters bind to different types of receptors. Dopamine, for example, is a neurotransmitter that can bind to at least 5 different gates. These different receptors are distributed differently in different brain regions. So dopamine in the prefrontal cortex will have a different effect than dopamine in the striatum, for example. Most neurotransmitters are pretty small, and your brain can synthesize them fairly easily as long as you eat a healthy diet that includes protein. You see, most of them are just amino acids, plus or minus a few bits and bobs. For example, the neurotransmitters you probably hear about the most are dopamine, norepinephrine, and epinephrine, or adrenaline. These are all monoamines because they only contain one amino group, and they are all derived from the amino acid tyrosine. Serotonin and melatonin, 2 other monoamines, are derived from tryptophan, and they play a role in regulating sleep, among other things. You might know that tryptophan is found in turkey meat. And here we come to another myth, that you're sleepy after Thanksgiving because you've just eaten food laced with the precursor to the neurotransmitters that make you sleepy. Well, next time someone shares that fact with you, let them know that chicken has a higher concentration of tryptophan than turkey. Overindulging in food is what probably makes you sleepy, if not the scintillating conversation at the dinner table and the emotional exhaustion of being with family, and at our table, the freely flowing red wine. But let's get back to the myth at hand, that mental illness can be simplified into a chemical imbalance of neurotransmitters. What I'd like to do for the rest of this lecture is to explore 2 disorders that have both benefited the most from the rise of pharmaceutical treatments and have proven to be far more complicated than their treatment success promised: schizophrenia and depression. Now, before we get too deep into a discussion of mental illness, I just want to take a moment to make a quick public service announcement. There remains a fair amount of big stigma associated with mental illness that is not present in our treatment of patients with other diseases. So I try to address this stigma whenever I talk publicly about such patients. And while there are many ways in which the stigma permeates our interactions, one way that might help is to be conscious of our vocabulary. Many of the patients that I have worked with who suffer from a mental illness resent that they are often labeled by their illness in a way that other patients are not. We don't talk about someone with breast cancer as a canceric, or someone with heart disease as a cardiopulmonaric. But we do call people schizophrenics, epileptics, and depressives. Their diagnosis is intertwined with their very identity as people. Or arguably, we see the disease, not the person behind it. We've made strides in how we talk to and about people with disabilities by putting the word people first. And while some patients don't mind, others have told me that they would prefer to be called a person with schizophrenia or epilepsy or depression. People first, disease second. A small thing, but maybe not such a small thing in the long run. Okay, with that important side note in mind, let's examine schizophrenia. The very term schizophrenia is often misinterpreted to mean split personality. When someone does something out of character, their behavior is sometimes described as schizophrenic. Not only is this use of the word offensive to individuals who suffer from the disease, it's also simply wrong. Schizophrenia was originally coined to describe the disordered thinking that can accompany the disease. Split mind, not split personality. Schizophrenia describes a disease that is extremely heterogeneous. Two patients with the same diagnosis can present with very different symptom profiles. One might suffer primarily from what we call positive symptoms, an increase in the frequency or magnitude of certain behaviors, like delusions and disordered thinking. Another patient with the same diagnosis might show mainly negative symptoms. That is, an absence or a drop in frequency of certain behaviors like catatonia, which is a decrease in motor activity, or flat affect, a reduced ability to express emotions. These patients might stay in the same physical position for hours and show what we call waxy flexibility. When moved into a new position, they take up that position and remain immobile until they become exhausted. Or they might avoid eye contact and show no emotion on their faces. The patient with positive symptoms who's hearing voices and believes that the government is reading her thoughts will look superficially very different from the patient who is completely still and mute. And yet these two manifestations of the disease have some underlying things in common. Schizophrenia runs in families, and the same genes can be expressed in one carrier as mainly positive symptoms and in another as mainly negative. And the same patient can show more positive symptoms during one episode and more negative ones during another. But the relative frequency of symptoms is also an indicator of how much help drugs could be for a particular patient. That's because the serendipitous discovery of the drugs that are largely used to treat patients with schizophrenia was related to the treatment of positive symptoms, hallucinations, delusions, and some aspects of disordered thinking. And as I've already mentioned, the discovery of antipsychotic medication revolutionized the treatment of people with schizophrenia, shifting away from long-term institutionalization and towards a combination of psychotherapy, social work, and medication. Because antipsychotic meds can have such a dramatic effect on the positive symptoms of schizophrenia, neuroscientists proposed a theory of the disease based on how they understood the mechanism of action of these drugs. Most antipsychotic medications bind to dopamine receptors, blocking them from the dopamine that the brain produces. So the dopamine hypothesis of schizophrenia, as it's known in the field, suggests that the disease results from too much activation of a specific set of dopamine receptors called D2. Since dopamine receptors are found all over your brain, and different circuits of your brain have different functions, the dopamine hypothesis suggests that positive symptoms are a result of too much dopamine in one pathway or circuit, and negative symptoms are caused by too much of it in a different pathway. It's possible that even the distribution of these receptors across the brain is different in individuals with schizophrenia, explaining one of the ways in which the genetic factors might be operating, or the way that the genes are expressed, since trauma and hormonal shifts in adolescence might also make a person more likely to develop the disease. So it's not just about the amount of neurotransmitter in the brain that causes symptoms. How many receptors there are also makes a difference, and receptors can be built or lost through gene expression, hormonal signals, or traumatic experiences. But notice already that proponents of the chemical imbalance myth are in trouble here. Combating schizophrenia is not as simple as depleting the brain of dopamine. Where the dopamine is excessively activating a certain type of receptor matters. And drugs that block dopamine receptors have not been very successful in alleviating negative symptoms. Even so, the dopamine hypothesis has other data in favor of it, data that seem to bolster the evidence drawn from the treatment of the positive symptoms of schizophrenia. For example, drugs that mimic dopamine, when taken in excess, particularly over long periods of time, can cause psychosis, or symptoms that resemble those found in patients with schizophrenia. This is true of drugs like cocaine and amphetamine, but also of levodopa, used to treat patients with Parkinson's disease, which is caused by a loss of dopamine-producing cells in the substantia nigra. So patients with Parkinson's disease don't have enough dopamine circulating in their brain. Giving them levodopa, a precursor of dopamine, can help for a limited period of time. But too much L-dopa can cause symptoms that look similar to the positive symptoms of schizophrenia. Considering all of this evidence, the idea that schizophrenia is caused by too much dopamine sounds compelling. But not only is the dopamine hypothesis more nuanced than just a proposal that there's just an imbalance of one type of neurotransmitter, there's also emerging evidence that dopamine might not even be the main player. For one thing, many patients don't respond well to drugs that block D2 receptors. In one study, even when 90% or more of D2 receptors were blocked in some patients, they still failed to show improvements. Another line of evidence comes from atypical antipsychotic medications. These are newer drugs that cause fewer side effects. In particular, Parkinsonian symptoms seem to be less prevalent with these new drugs, and they're more effective than the typical antipsychotics in treating the negative symptoms of schizophrenia, but just as effective at diminishing the positive symptoms. The atypical drugs, though, have been found to be less efficient at blocking D2 receptors, according to some studies, and they also affect serotonin, another neurotransmitter. The bottom line is that they don't have a simple mechanism of action. What's more, glutamate, the most common excitatory neurotransmitter in the brain, has also been implicated in schizophrenia. Drugs that block glutamate receptors, like ketamine and angel dust, can also induce schizophrenia-like symptoms. Nicotine increases levels of glutamate in the prefrontal cortex and is commonly abused by patients with schizophrenia, who report that it helps them focus. So scientists studying people with schizophrenia have put forth a glutamate hypothesis of the disease. They suggest that glutamate keeps dopamine in check. Without enough glutamate, dopamine has a larger influence on brain function than in a healthy person, and symptoms of schizophrenia might be the result. But drugs that increase glutamate levels in patients with schizophrenia have not been very successful at reducing positive symptoms. And, to complicate matters further, neurotransmitter levels aren't the only ways in which the brains of people with schizophrenia are different from those of comparatively healthy people. Volumes of brain matter in certain regions, like the frontal cortex and the temporal lobes, are smaller in many people with schizophrenia. Functional differences, that is, the way in which certain tasks activate the brain, have also been found in regions like the frontal and temporal lobes and the hippocampus. For example, in patients with schizophrenia, the frontal lobes are activated differently during certain cognitive tasks, like keeping a series of digits or letters in mind using working memory, compared with healthy individuals. and the connections between regions also seem to be different in patients versus controls. It's not clear whether these neuroanatomical and functional changes are a cause or an effect of the disease, or even the medications themselves. And we don't yet know what is causing the changes in neurotransmitter efficacy. What is clear is that schizophrenia is a complex disease with many factors and many different prognoses. Simplifying it as a chemical imbalance is not at all accurate, nor is it helpful. And yet some drug companies have been accused of doing just that as a marketing tool. Patients with schizophrenia are not alone in being targets of pharmaceutical company advertising, of course. Arguably, a far greater market is patients with depression. And here, too, we see the chemical imbalance myth tossed around liberally. The chemical imbalance theory of depression has its roots in the serendipitous discovery of tricyclic antidepressants like imipramine. We now know that tricyclic antidepressants work on a class of neurotransmitters called monoamines. Dopamine, serotonin, and norepinephrine are all affected by the drugs. Around the time that imipramine's antidepressant properties were being touted, another group of physicians published their finding that a drug designed to treat tuberculosis seemed to produce euphoria in some patients. This drug, too, worked on monoamines, inhibiting the enzyme that breaks them down. This enzyme is known as monoamine oxidase. As less of a neurotransmitter is broken down, more of it is available for use by neurons. So administering a monoamine oxidase inhibitor leads to more norepinephrine and serotonin floating around the brain. Many antidepressant drugs act on norepinephrine, dopamine, and serotonin, but each neurotransmitter is involved in a different set of brain functions. Norepinephrine modulates attentional focus and general arousal levels. Dopamine is involved in feelings of pleasure, motivation, and working memory. Serotonin can decrease anxiety, obsessive rumination, and compulsions. Every one of these functions is implicated in people with depression to different degrees. So psychiatrists are counseled to create a recipe of drugs that match the symptom profile of a specific patient. But what does this tell us about the idea that depression is just a chemical imbalance? Well, if that were true, you'd think that symptom abatement would happen as soon as the chemistry is reset, right? It doesn't take much time, after all, for me to experience the psychological effects of a cup of coffee. Basically, as soon as it's crossed the blood-brain barrier, I'm feeling pretty energized. But with antidepressant drugs, there's a therapeutic lag. The neurotransmitter levels are altered almost immediately, but it takes weeks for patients to feel better. Why? It's a big conundrum. But it means that it's not just about restoring chemical balance. What's more, it's not like patients with depression have low levels of monoamines to begin with. Many patients, even with major depression, have the same levels of neurotransmitters in their cerebrospinal fluid as their healthy counterparts. And lowering levels of monoamines in healthy people doesn't lead to depression. And some patients experience symptom relief from antidepressants that don't even act on monoamines, at least not directly. And cognitive behavioral therapy, during which patients work with a therapist to change thought patterns and reactions to negative situations, can be just as effective as drug treatments for people with mild to moderate depression, even showing similar neuroanatomical and physiological changes. Perhaps putting the nail in the coffin of the chemical imbalance story of depression, many patients with mild to moderate depression show as much improvement when taking a placebo as they do with pharmacologically active drugs. In fact, one article from 2011 in the British Journal of General Practice argues that it's more ethical to try a placebo treatment for patients who don't have access to cognitive behavioral therapy before prescribing SSRIs. Just like in patients with schizophrenia, there's a long list of other physical differences in the brains of people with depression. Even the way the brain changes in response to experience, what we call neuroplasticity, is different in some patients. For example, stressful events have different effects on the wiring of the hippocampus in patients with depression compared with healthy people. So I hope that I've succeeded in convincing you that the idea that mental illnesses are simply a function of a chemical imbalance is far too simple to be accurate. But of course, we do alter the balance of chemicals in our brains all the time, sometimes to predictable effects, such as when you drink a cup of coffee or a glass of wine. So where do we go from here? Do we just toss out drugs and start from scratch? Of course not. The trend now in the pharmaceutical industry is to replace the sledgehammer that's been prevalent in the past with a more delicate instrument, a scalpel perhaps. One example of this new approach is the use of genetic information to find designer drugs, tailoring them to the patient in much more specific ways. But we should also consider the psychological treatment options, seeing as cognitive behavior therapy can be so effective. Finding other non-invasive ways to rewire and reshape the brain with therapy is another promising future direction. But it's time to retire the idea that mental illness stems simply from a chemical imbalance, because so much nuance is missing from that explanation. And by reducing complex illnesses to what is becoming a cliché, we miss opportunities to find more effective treatment.